In this video, we are going to teach you how DNA replicates. DNA replication occurs over a variety of steps in a manner so that as few mistakes occur as possible, so the correct genetic material can be passed on during the cell division cycle. One thing to note about DNA replication is that it is considered semi-conservative, which means each parent DNA strand acts as a template for a new strand. So when the two new DNA strands are created, each DNA will have one parental strand and one new strand.
DNA replication takes place in two basic steps, which we will get into further detail later. First, the DNA double helix is unwound to separate the two template strands. Second, the new nucleotides form complementary base pairs and are covalently linked together by phosphodiester bonds, which is known as a phosphodiester linkage. These nucleotides are added from the 3' end to the 5' end of the DNA, where a free hydroxyl group is located. DNA replication starts with the action of a protein complex binding to a specific site on the DNA molecule, typically referred to as the origin of replication, ORI for short. This protein contains several different enzymes, including DNA polymerase, to catalyze the addition of nucleotides as the DNA chain grows. The DNA polymerase needs a short starter strand, known as a primer, to begin this process. The DNA polymerase elongates a polynucleotide strand by covalently linking new nucleotides to these strands.
The primer is typically a short strand of RNA that is complementary to the DNA strand and is synthesized one nucleotide at a time by the enzyme primerase. The DNA polymerase in this replication complex can then add nucleotides to the 3' end of the strand until the strand is complete. The replication complex causes the DNA to separate, forming two replication forks. The replication forks open in one direction, and due to the anti-parallel nature of DNA strands, one of the strands must originate at the 3' end, and the other must be synthesized backwards. The strand that originates at the 3' end, the leading strand, can grow continuously as the replication fork opens. However, the one that doesn't start at the 3' end, the lagging strand, must be synthesized in a way that it grows away from the replication fork.
The lagging strand must be synthesized by short stretches that start toward the 3' end and go towards the 5' end. They are not a continuous strand like the leading strand is. These fragments are known as Okazaki fragments, and there are many of these fragments along the lagging strand that each must be synthesized by its own primer. This causes gaps between the fragments, which are bonded by DNA ligase, an enzyme that makes the lagging strand's fragments a continuous strand. After the lagging strand and the leading strand are synthesized, telomeres play an important part in making sure the DNA strands do not join to other chromosomes by forming strings of repetitive sequences. These sequences ensure that the ends won't join together. Some cells eventually lose the telomeric DNA, but the enzyme telomerase ensures that the cells will be able to keep replicating by catalyzing the addition of any lost telomeric sequence.
Hey, thanks for watching. Now you know how DNA replicates.